Hey everyone, my name is Mark Morrow. Today we'll be talking about the infectious disease known as SARS. SARS stands for Severe Acute Respiratory Syndrome. It is named that because the disease mainly affects your respiratory system. Disease was initially spread in November of 2002 by a coronavirus now known as SARS coronavirus or SARS-CoV. In 2019, SARS resurfaced caused by another coronavirus called SARS coronavirus 2. Instead, this time, it wasn't called SARS, the disease was instead called COVID-19. SARS coronavirus and SARS coronavirus 2 share a 79% similarity in genomic structure. Both share a bonding protein receptor called ACE2. SARS coronavirus is called, or usually said to be like a large virus. It's positive single-stranded RNA virus. It is a helical nucleocapsid, and it is 29.75 kilobases in measurement. The 3' terminal region is what encodes for the protein specificity of it. So we're talking about the specificity of the surface spike, abbreviated as S, nucleocapsid, abbreviated as N, the membrane, abbreviated as M, and envelope protein, abbreviated as E. Usually these abbreviations are used referring to this virus in articles, just to kind of shorten, stop saying the word nucleocapsid and say N a little bit quicker, I guess. ICTV taxonomic classification for the coronavirus starts with realm being Riboviria. The phylum is Incertae. The order is Nidoviralis. The family is Coronaviridae. The genus is Beta coronavirus, and the species is human. So coronaviruses get their name corona from corona, meaning crown in Latin. It's called crown because of the glycoproteins on the surface of the virus. Those glycoproteins kind of give it a crown-like surface, so we call it coronavirus because underneath the microscope it's like a crown. They usually target the respiratory system of your body. It may target other parts, but usually just your respiratory system. They are of zoonotic origin, meaning they transferred via animal to human, usually from bat to human. The most notable coronaviruses, maybe you've heard of these, so SARS coronavirus, meaning the agent causing SARS, SARS coronavirus 2, the agent causing COVID, and then MERS coronavirus, I'm not sure if you've heard of that. It was outbreak in 2012, MERS meaning Middle Eastern respiratory syndrome, 2012, but it was camel to human instead of bat to human for that one. And then I'm not sure if you heard of neo coronavirus. The mode of transmission for this coronavirus is primarily it's transmitted by respiratory droplets, so inhalation of infectious droplets from another person. So you inhale it and then it infects you. Direct contact with infectious secretions, and an exposure to contaminated fomites. Fomites being the inanimate surface that the virus may cling to, and then maybe you touch that surface or somehow that virus comes off that surface and you inhale it. I talked earlier about the ACE2 receptor. This specific ACE2 receptor is important because it's how the glycoproteins on the virus enter into your host cells. The glycoproteins bond to that ACE2 because it's a binding protein receptor, and then it enters into your host cell. The virus mostly fluctuates to the lungs, small intestine, and kidney. This is thought to be because of the high density of ACE2 receptors in those cells, in those organs, but it's not known for sure whether that's the case. The main portion that is targeted is the alveolar epithelium of your lungs. The function eventually leads to pleural effusion, pulmonary edema, and condensations with relative sparing of the upper respiratory tract. This response to the infection is most likely linked to an overinflux, so too many cytokines are being released. So the area of infection causes the inflammation, and this is what is thought to be why your respiratory system responds in that way. And here I talk about these spike proteins of the virus. The N-terminal contains the receptor binding domain, and the C-terminal portion contains a membrane-anchored subunit. The coronavirus enters into your body in 3 stages, the first being receptor binding, second being conformational change in S protein, and then third being cathepsin L-mediated proteolysis. So first, the incubation period is between 2 to 10 days. Sometimes it ranges a little bit higher, and sometimes the incubation could even be 1 day. On average, it was about 4 days long, however. Symptoms were started out as being flu-like. So you had a fever, so what is that, 37 Celsius, over 37 Celsius for your body temperature. You had chills, shivering, you had a headache, malaise, which is like general feeling of discomfort when you had this disease, and amalasia, which is muscle fatigue, muscle soreness. And then in 10 or 20% of cases, diarrhea was also a symptom. 2 to 7 days later, you would have a dry cough. So here's where like respiratory problems come into play. You have a dry cough, dyspnea, which is like a shortness of breath. And the shortness of breath leads to hypoxia, which is low levels of oxygen, low levels of blood oxygen. In 10 to 20% of cases, they got severe enough to where you require a ventilator to breathe. And if these respiratory problems were like too harsh, in 9.6% of cases, it would lead to the death of the patient. You could diagnose SARS at the time 3 ways. First being RT-PCR, which stands for reverse transcriptase polymerase chain reaction. And this is basically just testing for the virus itself, the coronavirus, the SARS coronavirus. The second being seroconversion by enzyme-linked immunosuppressant assay, or indirect fluorescent antibody. This was mainly testing for the antibodies to the SARS coronavirus that may have been present in your host cells. Third being isolation in cell culture of SARS coronavirus using RT-PCR as a confirmation. So this would be putting tissue or blood that was suspected to be infectious into a container with cell culture, and then you would examine that cell and see if it had been infected, and you could do that by using RT-PCR as confirmation. I did want to mention here that the RT-PCR test usually was administered with a swab. I'm not sure if you guys remember this during the peak COVID, or maybe you've had a COVID test recently, but you'd have that shoved into your nose as far back as you possibly could to get it to test up here in the nasopharyngeal part. And that's where you could test for whether the virus was there or present. Sorry. No vaccine was ever designed for SARS coronavirus because it kind of been very quick. So it got about over the course of 6 months, it had the outbreak had finished. So no vaccine was ever really needed. The host was instead supported by different factors such as ventilators to assist breathing, antibiotics to kill the bacteria that cause pneumonia, or steroids to reduce inflammation in the lungs. Here I talk about the development of the SARS coronavirus, and I said there's no concrete answer on where the coronavirus developed, but there are a couple of main theories or ideas of how this virus was developed. The first was the natural host was wild bats. Little bat down here on the bottom left. On the bottom right, I have a civet. This was another idea of who the actual natural host was for this coronavirus. It was thought to be first transmitted via the wet markets of Guangdong province, which is a province right outside of Hong Kong in China. And it was thought that maybe someone had eaten one of these animals that had the coronavirus in it. Here's a little run through of timeline of the outbreak of the coronavirus of SARS. It starts with the atypical pneumonia. It's first documented in Guangdong province. So usually it's just it was called atypical because of its fever-like symptoms. And then on February of 2003, the next year, the World Health Organization received reports of a respiratory disease. A little later, a doctor from Guangdong who's had the disease stays in a hotel and spreads a respiratory disease. So 2 things on this part. This was mainly how the disease had been spread so well. It was because it was a nosocomial spreading. So that means that because of the doctor, he was the person dealing with the infection. It was like hospital transferring because he'd become common contact with that. And the second thing was that this was also known as one of the super spreader events. This is how the virus spread so well, was because of this right here. He's in a hotel and he gave it to a few people in the hotel who had transferred it to Vietnam, Singapore, and Toronto. And then from there, it kind of just kept growing pretty quickly. In March, the World Health Organization issued a global report or global alert of this outbreak. A little later, over 150 cases are now reported disease, and they now call this disease Sars. World Health Organization starts the study of SARS causation a little later, and then the coronavirus is now identified in patients with the disease. On April 12th, the genome of the coronavirus is completed, and then a few days later, the World Health Organization announces the coronavirus linked to SARS, and they call it SARS coronavirus. June 5th, this is the absence of future transmission, indicates the end of the outbreak in humans. So that's the official end, or I guess maybe unofficial end of the outbreak. Here's some stats relating to the actual outbreak. SARS between 2002 and 2003 was responsible for 774 deaths. It infected over 8,000 people. The disease spread over 5 continents to 29 countries, and the fatality rate was 9.6%, so about 10%. Most people who were infected were actually healthy adults between the ages of 25 and 70, and surprisingly, there were a few cases with children, because usually children are more susceptible because of their weakened or undeveloped immune system, making them more susceptible. But a few cases were identified with children under the ages of 15. Here I have a future outlook on, the future outlook on what to think about how we respond to this, I guess. And I have a little quote from NCBI. It said, "An important lesson learned in China was the need for transparency and communication. SARS was a frightening reminder of how quickly a novel infectious disease can emerge and spread. Humankind has had a lucky escape. Suppose that SARS coronavirus became readily transmissible from person to person some days before people became seriously ill with disease." So basically he's saying here that, that because it had been spread only when you had symptoms, that we kind of got lucky with that portion, because it did have a high mortality rate, 10%. And he said that in the future, how we respond better to this would be with transparency and communication. He in the article, he mostly called out China for being a little bit precautionary of how they withheld information from the World Health Organization. So he's saying in the future to combat this much quicker and not let this get as bad as it did, to be more transparent. All right, I think that's going to do it. Here are my resources. Thank you so much for watching the video. Have a great day.